Again, I'd like to thank everyone for joining us at the Frontiers in Child Health Research Seminar. The seminar is held on the second and fourth Mondays from September through June and includes speakers with work focused on child health from both UCSF as well as guest speakers. Please let me know via email if you're interested in speaking or would like to suggest a speaker. Looking ahead to our next speaker, we'll be welcoming Dr. Matt Pantel from the Division of Pediatric Hospital Medicine on May 8th. We're still virtual and using Zoom, and we ask everyone to stay muted unless you have a question, and we'll also open it up to questions at the end. Today, we're welcoming Dr. Jason Boehme from the Division of Pediatric Critical Care. Jason's an assistant professor of pediatrics. His research is focused on metabolic perturbations associated with pulmonary hypertension, pulmonary vascular disease, and abnormal lymphatic pulmonary flow. His research is funded by NHLBI. The title of his talk today is Activation of Hypoxia Inducible Factor Signaling by Abnormal Hemodynamic Forces in the Pulmonary Vasculature. Take it away.
All right, let me share my screen. And here, okay, is everyone seeing my slides?
Yes.
Okay, great, thank you. Okay, so I will talk about pulmonary vascular disease today. So I just wanted to kind of, and with the audience, there's some people here who have a lot of expertise, and obviously some people who are totally unfamiliar with kind of what I'm doing. So just to briefly talk about the clinical context, I wanted to touch on a couple of things. So this is a registry study from several centers in North America that was published relatively recently. And I wanted to highlight a couple of aspects of pulmonary hypertension and pulmonary vascular disease in children. From the pie graph on the left, the first thing you can kind of see is that this is kind of a multifactorial and heterogeneous disease that is caused by a number of different sort of underlying driving forces, many of which kind of get lumped under the common umbrella ultimately of pulmonary hypertension. But even within these groups, you can see the table on the far right is just breaking down the subgroups within group 1. There are really many different kind of conditions and risk factors that predispose to pulmonary vascular disease and pulmonary hypertension. In the survival curves in the middle, you can see that within these groups the outcomes are quite disparate depending on the etiologies, but that overall, unfortunately, there still is quite a high degree of significant disease, mostly resulting in death, although with some lung transplantation, actually growing population of lung transplantation within the lung transplant field. So specifically, I, as a fellow, joined Dr. Feynman's lab and have worked with him and Emin Maltepe from the Division of Neonatology and Sanjeev Datar, also in critical care, looking at the pulmonary vascular disease in congenital heart disease. And there's a couple very interesting aspects of this group of pulmonary vascular disease, and I like this cartoon. It shows a couple of kind of nice aspects to studying this particular subtype. The first is that of all of the types of pulmonary hypertension, this one is extremely well characterized relatively in terms of both the clinical progression as well as the histopathologic progression of the disease, and you can see that in the cartoon, where earliest you kind of have development of endothelial physiologic dysfunction, and as you start to develop more advanced disease, you start to get actual vascular remodeling, first with sort of distal muscularization of the pulmonary arteries, and start to recognize thickening of the smooth muscle medial layer of the pulmonary arteries. And then as the disease gets more severe, you start to get more complex vascular neointimal lesions that involve proliferation and sort of abnormal growth of a number of different vascular cell types, not just the endothelium, but also the smooth muscle cell, fibroblasts, and also abnormalities of inflammatory cells within the vasculature. And ultimately losing vessels with very complex obliterative lesions. Another aspect that I've always found very fascinating about this setting of disease is that it's been recognized for a long time that in children with congenital heart disease, there's a certain inflection point where surgical repair of the heart disease will lead to clinical reversion and reversal of the pulmonary vascular disease, as far as we can tell. But once you hit this kind of inflection point, even if you have a surgical correction of the underlying heart disease, the vascular disease itself will not only persist, but will continue to progress and get worse. And it's quite clinically relevant as well, because those patients do very poorly, even within the subgroup of congenital heart disease and pulmonary vascular disease. The unfortunate aspect is we actually are pretty rudimentary in understanding when and why that point of irreversibility happens. This is a slide I stole from Jeff that is in many of his talks. But really our ability to know when this vascular disease becomes irreversible is pretty rudimentary, and can largely be broken down by age or time of exposure. And then the other risk factor really has to do with the nature of the hemodynamic abnormalities. And to kind of, summarize this a little bit, although it's much more complicated, is that lesions like truncus arteriosus or complete AV canal, where the pulmonary vasculature receives not only high flow, but also, very high pressures of communication with the systemic circulation, those patients are at highest risk of developing irreversible vascular disease, and it will happen most quickly in those types of patients. Down at the bottom of the top box is an ASD, a lesion with increased flow, but at low pressure and variable increased flow. And there, the chance of developing pulmonary vascular disease is quite low relatively to other lesions. And when it does happen, it tends to be much later in life, to the point that, this is, often considered more of a kind of additional risk factor in the development of pulmonary vascular disease in these patients, rather than a causative, singular etiology. So Dr. Feynman and Dr. Reddy, now about 30 years ago, developed kind of the original large animal model of congenital heart disease that we still use. I'll mostly be talking about the Shunt model, but I will mention briefly one or two other large animal models that we use to study vascular disease and vascular biology in this setting. So the surgery is in fetal sheep, and while they're in utero, we work with the CT surgeon up at UC Davis to externalize the fetal lamb still on the placental circulation, and basically anastomose a large graft between the aorta and the pulmonary artery. Then the uterus is closed, and the fetal lambs are birthed usually 1 to 2 weeks after the surgery. And in utero, this is very much like a ductus arteriosus. It doesn't change the fetal circulation. However, after the animals are born and the ductus closes, they're left with this large, persistent communication between the aorta and the pulmonary artery that ultimately will result in high pressure and high flow into the pulmonary circulation. And then we study those animals at various ages, although predominantly when they're at about 4 weeks of life. And we use different methods to kind of study the vascular changes, which I'll talk about as we go. The kind of one of the earliest hallmarks and still a major biologic and physiologic aspect that we're looking at these has to do, again, with that early endothelial dysfunction in patients with congenital heart disease. And so this is from early work looking at vasodilator testing. So actually, anesthetizing the animals in vivo, giving them various vasoactive substances. And the two that I'll highlight are acetylcholine. So that's a vasodilator. It acts at the endothelium, causes the endothelium to produce nitric oxide, and the nitric oxide then goes to the smooth muscle and directly relaxes the smooth muscle and relaxes the vasculature. Alternatively, we can just give inhaled nitric oxide and bypass the endothelium altogether to directly act at the smooth muscle. And then on the right is some vasoreactivity testing that, again, was done early on in the shunt animal. And what you can see on the left is looking at acetylcholine, is that in the controls, when they're given acetylcholine acutely, you see a percentage decrease in the pulmonary vascular resistance. That's the white bar. As opposed to the shunt animals, when they're given acetylcholine, have a much different response. And in this study, there's actually a slight increase in the pulmonary vascular resistance, although, over many studies, what we see is definitely a decreased vasodilation in response to this endothelium-dependent vasodilator. However, over on the far right, when we give the animals inhaled nitric oxide, there's no statistical difference, indicating that with adequate nitric oxide, the smooth muscle cells can relax the vessels, but it's really the endothelium that's having the primary deficit on this testing. So again, this is from work that was done a while ago, and this is kind of a busy slide, but what I wanted to highlight here is that a major kind of physiologic reason for this endothelial dysfunction has to do with the production of nitric oxide by the endothelium, and that is primarily mediated by endothelial nitric oxide synthase, or NOS or eNOS. And the data here is kind of showing very nicely that what happens is that the enzyme eNOS is present and even a little bit increased in the shunt animals, and is actually quite active. So when you look at enzymatic activity in the shunts compared to the controls, so that's the black bars mostly compared to the white bars here, is that the longer the animals have this abnormal blood flow, this enzyme activity is increasing. However, in kind of the middle column, what you see is that that increased enzyme activity is associated with decreased production of nitric oxide, which is measured both in the plasma and in the lung tissue. And then on the far right, showing that that corresponds to an increase in superoxide production. So this characterizes a phenomenon of eNOS called uncoupling. And what this has to do with is the normal domain of the eNOS protein usually will take oxygen and arginine and produce nitric oxide. However, when there's disruption of the many cofactors of the enzyme, or when it is in a monomeric as opposed to a kind of a dimeric configuration, that oxygenase enzyme will convert oxygen into superoxide radicals, which will go on to have various biologic effects. And so that's what's called uncoupling, and a very common hallmark of the endothelial dysfunction that we see in this model. So just kind of briefly going back, this sort of puts our model, I'm not showing the data, but there is very early signs of smooth muscle hyperproliferation and medial hypertrophy in the shunts. But primarily at this early phase of endothelial dysfunction, which again makes it somewhat unique in terms of animal models, that it's capturing a very early phase of pulmonary vascular disease progression. So what I'm going to talk about today is kind of an ongoing project that is very interesting, and some of this is published in a paper with Sanjeev Datar, which I'm showing here. And so from this model, we've isolated various cell lines, and this is all going to be focusing on endothelial cell lines that we have grown out from these animal models. And this is actually looking at lymphatic endothelial cells. So this is from the efferent vessel of the caudal mediastinal lymph node, so kind of a common lymphatic vessel from the pulmonary vasculature. And when we isolated endothelial cells from this lymphatic vessel, we sent the cells for bulk RNA sequencing. And on the left, you can see some of the enrichment analyses. And some of the things we saw were, quite intuitive. We see responses to mechanical stimulus. We see cellular responses to fluid shear stress, which is what we expect in the setting of increased blood flow and increased lymph flow. But then the one that's, very interesting and a little bit unexpected is the cellular response to hypoxia. And when we looked at that more closely, a lot of that was actually driven by HIFs, which are hypoxia inducible factors. And on the right here, you can see sort of a network mapping image that displays the direct and indirect targets of HIF-1 alpha, which I'll talk about a little bit more, or rather a lot more. This is a very similar analysis looking at pulmonary artery endothelial cells, so now endothelial cells from the pulmonary arteries themselves. And same process, sent for bulk RNA sequencing. And what you can see here, this is from a different program. This is from IPA, but a very similar analysis showing enrichment within hypoxia signaling pathways and enrichment related primarily to HIF-1 alpha, although with potentially some of the other HIF alpha isoforms involved. And on the right, again, a network analysis showing a lot of the direct and indirect HIF-1 transcriptional targets that are upregulated in the PA endothelium. So this talk is going to be about primarily HIF-1 alpha. So to sort of briefly touch on that, it's named based on the original descriptions as the hypoxia inducible factor, which is very fitting. And the regulation of HIF-1 alpha is quite interesting in the HIFs in general. It's largely not at the transcriptional level, but actually at the protein stabilization level. So HIF is primarily regulated through kind of a targeted destruction mechanism where under normoxic conditions, prolyl hydroxylase enzymes hydroxylate residues within the HIF alpha subunits. Those are then recognized by von Hippel-Lindau, which ubiquinates HIF-1 alpha, and that is then targeted for degradation. Under hypoxic conditions, what happens is there's inhibition of the prolyl hydroxylases. So the HIF alpha subunits are stabilized and maintained at higher levels, and then they can bind to HIF-1 beta or ARNT and other transcriptional cofactors, and then they associate with hypoxic responsive elements in the promoters of many different genes and upregulate transcription. So HIF is extremely important in kind of vascular biology and in angiogenesis. And this is just kind of a cartoon showing, the kind of original descriptions of HIF, where low oxygen environments stimulate HIF, creating targets of pro-angiogenic molecules. This is showing VEGF, obviously very important in angiogenesis, that then stimulates, development of new blood vessels. So, this is an extremely interesting target, and HIF is also very well recognized in pulmonary hypertension and pulmonary vascular disease. On the left is a picture I pulled off of Google of the Volga River. So there's a very interesting population of patients with this disease called Chuvash polycythemia, and Chuvash is a region in modern-day Russia on the Volga, where the inhabitants of this region carry, sort of a rare mutation in von Hippel-Lindau that's autosomal recessive, but this specific mutation inhibits the ability of von Hippel-Lindau to recognize and ubiquinate hydroxylated HIF, which results in inappropriate and high HIF signaling in multiple body tissues in the absence of hypoxia. And these patients become polycythemic because of HIF-2 alpha's control of the hormone erythropoietin, but they also develop pulmonary hypertension. So this is, extremely interesting. This is kind of the prototypical HIF and pulmonary hypertension association, but HIF is also widely implicated in many types of pulmonary vascular disease, most commonly in the types of pulmonary hypertension associated with chronic hypoxia and hypoxic signaling, but also stabilization of HIF under kind of normoxic conditions is also, widely described in advanced pulmonary hypertension from many different etiologies. And HIF is really a transcription factor that's very wide-reaching, and so depending on the cell type, it influences sort of cell proliferation, migration. It has direct influences on a number of growth factors, also on cellular metabolism, and then a number of different vasoactive and vasoconstricting substances. So many different ways it influences vascular biology in the setting of pulmonary hypertension. Now, the reason it's particularly interesting to see in our model is that our model is not hypoxic. And so the cartoon on the left is of a PDA, but the physiology of the shunt model is very similar in the sense that, what we see is shunting of highly oxygenated blood from the LV and the aorta into the pulmonary arteries. And so what you actually get is an increased oxygen tension in the pulmonary artery compared to normal. So, the mechanism of HIF stabilization in this model is not due to hypoxia. So then we have to look a little bit deeper and sort of look at how cells actually respond to environmental hypoxia, which is a whole field. And to very briefly summarize it, it is predominantly through the mitochondria. And there's been very elegant studies showing that cells without an intact mitochondrial membrane and mitochondrial membrane potential, cells cannot stabilize the HIF alpha subunits. And the proximal signal is generally agreed to be that the mitochondria responds to environmental hypoxia by modulation of reactive oxygen species arising from the mitochondria. Exactly where those are coming from and the nature of the reactive oxygen species is a little bit controversial. There's a couple of groups, but we tend to agree with the more widely held kind of belief that in the setting of hypoxia, the mitochondria respond with an increase in production of reactive oxygen species, largely seeming to originate from complex 3. Those reactive oxygen species then, interact with or signal to the prolyl hydroxylases, inhibiting their activity and resulting in the stabilization of HIF. So, taking that idea, then we look at, why we're seeing stabilization of HIF in our model, which doesn't have hypoxia, but does have sort of abnormal mechanical stimulation of the vasculature. This is, one of several papers I pulled because the images were quite nice. This is looking at smooth muscle cells, so a little bit different than what I'll be talking about. But what you can see here, going from top to bottom, are smooth muscle cells that are being exposed to varying degrees of a stretch stimulus. And then TMRM is a fluorophore that responds to mitochondrial membrane potentials, and mitogreen is just a mitochondrial fluorophore. And what you can see over on the right on the merged images is that under what would be considered sort of normal approximations of physiologic stretch, the mitochondria actually arrange themselves in these sort of linear branching networks. And once you start to deviate from that, either with stretch that is too little at the top or stretch that is pathologically elevated at the bottom, you start to get sort of disruption and fragmentation of these mitochondrial networks. So then looking back at the endothelial cells, and this is pulmonary artery endothelial cells from control 4-week-old sheep and matched shunt 4-week-old sheep. And this was actually done on twins, so twin pairs, one without the shunt and one with the shunt. And then this is just, staining again for mitochondria. And this was done by some collaborators of ours. And what you can see in each pair on the left, you see these kind of, again, nice sort of fibrillar networks of mitochondrial staining. And on the right in the shunt, much more fragmented mitochondrial networks. And just doing some kind of simple quantification, we can show that the dimensions of the mitochondria are altered, and then it can also kind of quantify the networks, and also see that those are extremely different in the shunt compared to the control animal. So we're seeing again disruption of sort of mitochondrial networks, and then looking at mitochondrial function, again, using TMRM, which is a fluorophore that fluoresces in proportion to the mitochondrial membrane potential. See that in the shunt endothelial cells, the mitochondria have a diminished mitochondrial membrane potential compared to the controls on the left. And then on the right, mitosox is a mitochondrial targeted fluorophore that fluoresces in proportion to reactive oxygen species. So we see that the mitochondria have a diminished mitochondrial membrane potential, and also are increasing the production of mitochondrial reactive oxygen species. So this is, very similar and kind of recapitulating the physiology that's seen at least in acute hypoxia that leads to the stabilization of the HIFs. So that gets us back to our endothelial cells. On the left, our endothelial cells from the lymphatic vasculature. And what you can see is when we look at HIF-1 alpha levels, and again, this is under culture conditions at room air, we see stabilization of the HIF-1 alpha protein. In the middle, see the same thing in the pulmonary artery endothelium from our cultured cells. And then when we look at peripheral lung tissue, we can even see the same thing there, that there's increased stabilization of HIF-1 alpha protein within peripheral lung taken from the animals. So wanting to kind of look at a proof of concept, we can take the control arterial endothelial cells and subject them to different types of mechanical stimulation. And on the left, we're taking pulmonary artery endothelial cells and subjecting them to either cyclical stretch or to kind of a high tangential shear, which are different types of mechanical stimulation for the cells. And you can see that both of those actually increase the production of mitochondrial reactive oxygen species. And this is done over 4 hours, so this is a relatively acute cellular response. The other interesting thing I'll point out here is that you can see that stretch and shear actually kind of provoke somewhat different responses within the cell, although they're both different from control. On the right, looking at lymphatic endothelial cells, I don't have the mitochondrial ROS, but when we shear those cells, see that, again, HIF-1 alpha starts to stabilize very quickly under these mechanical stimulating factors. So, this is very interesting that we're seeing really mechanical forces resulting in a very similar phenotype in terms of HIF stabilization, as is seen in sort of other more classic kind of pathologic stimuli that will cause upregulation of the HIF enzymes. The other thing I'll point out here, the reason why we have stretch and shear in the pulmonary arterial endothelium is that, it is subject to kind of a wider variety of mechanical stimulation, both, dynamic stretch of the vessels during kind of systole and diastole, as well as, kind of tangential shear forces from the high pulmonary blood flow. The lymph is a little bit more restricted. It doesn't have an active driving pressure, so is more subjected primarily to shear forces. So, this is something that, Jeff and the lab have been interested in a long time, is how these differential mechanical forces result in different, biological kind of outcomes. And so just kind of briefly mention one of the other large animals that's been developed by the lab is an LPA ligation model, where it undergoes a very similar fetal surgery in terms of timing and approach, but rather than placement of a shunt, the left pulmonary artery is just entirely ligated. And that results in an animal that then has basically all of the right ventricular flow going out to the right pulmonary artery. So significantly increased flow, but with a much lower pressure head than what we see in the shunt animals. So kind of interestingly, looking at that model, we see that in the model with just an increased flow to the vascular bed, we have a much different response in terms of the production of mitochondrial reactive oxygen species, as well as the stabilization of HIF. So just, again, showing that the kind of nature and magnitude of the mechanical forces becomes quite important for how the cells are responding, and kind of ties nicely back to what we know from the clinical correlation. So from this observation, there's a lot of interesting questions that arise. So just kind of briefly, some of the things that we're interested in here is sort of what is that axis that transduces mechanical forces to this kind of mitochondrial signaling that we see downstream. As I've mentioned a couple of times, is it pressure or shear or the combination that mediates these biological changes that we're seeing, and how do those overlap and diverge? I think another, very important aspect is, this is an early disease model, which, we believe is reversible by most metrics. So as we're seeing things at this early stage of disease progression, are these truly pathologic findings, or are they just adaptive findings? There's many circumstances where, you want your vascular beds to adapt and accommodate increased flow, like during states of exercise or other kind of high cardiac output states. And then somewhat related to that, are these changes truly contributory to the pathology of pulmonary vascular disease, or are these just kind of sort of knock-on phenotypes to more upstream driving factors like the mitochondrial disruptions that we're seeing? This is from just a nice study to kind of remind me to mention that the HIFs have a very important role in pulmonary vascular development and pulmonary alveolar development. This is a brief snippet from a very nice study using a BPD, an inflammatory BPD model, where they basically, inhibited prolyl hydroxylases and showed that by, reestablishing HIF signaling through inhibiting these prolyl hydroxylases, it partially rescued the BPD phenotype in terms of both vascular growth and alveolar growth. So, we have to be kind of careful just saying that, HIF signaling is a bad thing at, these early developmental stages. So the approach that we kind of started with, and that I'll mostly talk about here, is we decided to try to use pharmacologic agents to sort of knock down or affect this signaling pathway and see what happens. So we started again with our in vitro model and are treating the cells with MitoQ. So MitoQ is a derivative of coenzyme Q10, but it's conjugated to an agent that basically targets it directly to the mitochondria, where it then acts as an antioxidant. So it helps attenuate the production of reactive oxygen species from the mitochondria. On the left is some data from our pulmonary arterial endothelial cells in vitro. And what you can see is that treatment with MitoQ helps restore a more normal mitochondrial membrane potential, and also helps diminish that heightened production of mitochondrial reactive oxygen species. On the mid-right, you can see that that does indeed translate to decreased stabilization of HIF-1 alpha, at least in vitro. And on the far right, you can see the same thing in the pulmonary lymphatic endothelium, that treatment with MitoQ helps decrease that stabilization of HIF-1 alpha that we see in the endothelial cells. So then the next thing is we're kind of starting now with characterizing the vascular responses with the treatment of MitoQ. This is a set of experiments isolating resistance vessels from the pulmonary vasculature when we studied animals at 4 weeks of age, and putting segments of those vessels onto wire myographs. You can see the cartoon of the setup on the left. So we then use a pharmacologic agent, in this case norepinephrine, to constrict those pulmonary arterioles up to a high contractile state, and then progressively increase the exposure to the endothelium dependent vasodilator acetylcholine. So you can see along the x-axis there's increasing doses of acetylcholine. So what you can see here is in the on the blue line, you have very nice vasorelaxation and control endothelial vessel segments in response to acetylcholine. Up on the top in orange is the shunt, and again you see, as we expect, a very blunted release of tension and vasodilation in response to acetylcholine. And in segments from the sorry, I should go back and say these are now MitoQ treated animals, where we actually started daily administration of MitoQ to the animals within a day or two after their birth, and continued that for the full 4 weeks until we studied them. So in these daily treated animals with MitoQ, they actually have improved response to acetylcholine. Looking at now the in vivo physiology, so this is actually again in anesthetized animals, and delivering the agents directly into the pulmonary vasculature. When we treat them, and this is looking at our standard shunts that are vehicle treated compared to MitoQ treated shunts. On the left is the responses to acetylcholine, and the acronyms are for the mean pulmonary artery pressure. The middle is indexed left pulmonary artery flow, which is the Q. And then on the right is the indexed pulmonary vascular resistance. So that's what we're most interested in. And you can see that in the MitoQ treated shunt, again, we're having a greater relaxation of the pulmonary vasculature, as seen by the greater decrease in pulmonary vascular resistance in response to the endothelium-dependent vasodilator acetylcholine. On the right is sort of the same physiologic parameters with inhaled nitric oxide, so endothelium independence. And interestingly, we do see some slight changes in flow, but again, not a significant change in the pulmonary vascular resistance. So highly indicating, an improvement overall in endothelial function in these animals treated with MitoQ. This is then kind of starting to look at that nitric oxide production. On the upper left are cultured pulmonary artery endothelial cells using a DIF fluorescence. This is a fluorophore that fluoresces in proportion to the production of nitric oxide. So, this is just, I think, 1 or 2 cell lines, and we're working on repeating this experiment with more cell lines that we have cultured. But seems like there is an improvement in vitro cellular nitric oxide production in these MitoQ treated cells. Looking to the upper right, this is sort of a similar way to assess that, is culturing the endothelial cells and then sampling the media to look at the release of nitric oxide into the media. And you can see the 3 columns are control shunt and these MitoQ treated shunts, showing a partial rescue of that phenotype. Again, improved release of nitric oxide into the cell culture media. Looking in vivo, we see a very similar phenotype when we look at the plasma or the whole peripheral lung tissue on the bottom, where treatment with the MitoQ is sort of partially restoring this production of nitric oxide in these animals. So, this seems to be accounting for the improved physiologic response we're seeing when we treat these animals with MitoQ. So I will just kind of briefly mention, obviously, people have looked at this, although not as much as you might expect in other animal models, and again, primarily in animal models of hypoxia. And I just thought their findings here were very interesting. On the left, what they're looking at is the RV systolic pressure, so the hemodynamic assessment, and this is without any kind of vasoreactivity testing. This is just the baseline pressure. And then also looking histologically at the vessels for muscularization, so signs of smooth muscle proliferation and expansion of the arterial medial layers. And basically what they find is that MitoQ treatment in these sort of chronic hypoxia rodent models does not seem to halt the histologic evidence of vascular remodeling, and it does not lower the baseline RV systolic pressures. However, looking on the right, what they do find, which is quite interesting, is that MitoQ does actually seem to blunt the remodeling of the right ventricle that they usually see. So there's decreased RV weight and thickness in these chronic hypoxia, pulmonary hypertension models when they're treated with MitoQ. And then the part about this that really caught my attention is that in addition to the hypoxia model, they also did a purely mechanical model where they put a PA band on a cohort of these animals, which similarly pressure loads the RV and will result in RV hypertrophy over time. So then again, looking at the same indices of kind of RV hypertrophy, weight and thickness, what they see is, again, in a purely mechanical model, very, very similar to what we are using in terms of the inciting stimulus. They also see that MitoQ is actually effective in attenuating RV hypertrophy in that model. So I think that's very interesting as it relates to the work we're doing. And I think as they are pointing out is that the MitoQ here actually seems to be modulating probably redox biology and reactive oxygen species signaling within the ventricular myocardium as well. Also something that we're quite interested in. But again, seeing this kind of mitochondrial redox signaling access as an important aspect driving changes at not only the vascular level, but potentially at the myocardial level as well. So there's a lot of work ongoing, but I think I am going to stop there and thank many people from the Feynman lab group who have helped me over time. That was some metabolism data that I can show to people if they're really interested in those changes.
Thank you, Jason, for sharing all of that. I'm going to open it up to questions. Yeah, so I think just from that last piece you were showing, I was trying to sort through the figures, but it looks like the effect on the RV was then independent of the elevation in the downstream resistance, or
yeah,
is that not clear?
It is. It's not clear, but that is certainly what the authors postulated, is that MitoQ was probably affecting the mitochondrial ROS of the myocytes themselves that was meeting that signal. But I thought it was fascinating that they showed the same thing in a PA band model.
Right. That's
not just a
part
chronic
that,
hypoxia.
Yeah. because you wouldn't expect to have seen. Yeah, that is just interesting. Okay, well, I'll let Jim go ahead.
Hi, thank you. Very nice work. I might ask you a computational question dealing with the transcriptome data.
Okay.
So there is an approach to drug repurposing that's based on some really exceptional stuff developed out of the Broad called the connectivity map testing of. You're shaking your head. Do you know about CMAP?
No, I don't.
So testing a variety of cell lines against a variety of drugs at submicromolar levels, and you pair the transcriptome profile in your experimental model with what these drugs induced in particular cell lines, and you're looking for, in terms of a therapeutic drug, you're looking for something that would give a reciprocal to the upregulation and downregulation. Could chat offline. We have a local maven who's very good at, knows a lot about this, Marina Sirota, but it just comes to mind since this is a translational seminar and you're working on MitoQ and trying to find drugs. The optimal experiment might be using isolated endothelial cells and then doing the RNA-seq. But it's a thought. CMAP.
CMAP. Yeah, I will. I mean, I'll definitely check it out. It's something we're very obviously very interested in of developing potential therapeutic targets, and we've talked about using our cell culture model to do some kind of more high throughput screening types of experimentation, and so that would be great.
So CMAP was developed to that end. That's why all the drug concentrations used, and in each cell line in each experiment they did about 3 concentrations, but they were all submicromolar to put them in therapeutic range. That's a possibility.
Yeah, that sounds great.
Yeah, along those lines, I wonder, like, if you, the right ventricular hypertrophy is, right, it's compensatory, right? So if you gave something systemically, both to help the vasculature, right, but then potentially mitigate this compensatory mechanism, would you actually, be better or not? Like, I don't know. I guess I'm just thinking, like, just along those lines when you're thinking about testing something. Okay, Ron has his hand up first, and then
Jason, really nice talk. I'm wondering, given the codependency of pulmonary vascular development and pulmonary alveolarization on VEGF and nitric oxide, I'm wondering, I know this isn't your focus, but in your model, do you also see parallel changes in alveolar genesis in these animals?
Yeah, that's that is a very good question. To my knowledge, we do not, and I know, like, a long time ago, Jeff worked with Marlene Rabinovich, and then more recently we've done several kind of like histologic analysis with Chava Galambos at Denver, and never really seen it, never really seen any changes in alveologenesis. Interestingly, Sanjeev now is working with a lab up at UC Davis to actually do pulmonary function testing in the animals. So, I think he's getting ready to publish some of that data soon, which is quite interesting, because we definitely do see differences in the PFTs.
Thanks.
I think this has also come up with the ductal ligation model, for the fetal ductal ligation model, because the question, because a number of the modulators for pulmonary vascular disease, the pulmonary vasodilators, also do have trophic effects on the lung and alveolarization. So it's a good question. Yeah, I'll have to drop off, or
No, I'm still here.
Answered. Okay, go ahead. You still have questions.
Thank you. Yes, I do. Jason, it's fascinating to me that it seems like you actually get different results from shear versus pressure. And, I wonder if shear is, kind of somehow a more tangential force to the cell membrane, and pressure is more orthogonal or kind of perpendicular. But do you have a feeling for what cellular mechanisms? I mean, I'm thinking about in my mind's eye, I guess, like proteins somehow sticking out from the cell membrane. Like, what are the different mechanisms that respond to shear versus pressure?
Yeah, that is a great question, and it's something that Jeff has been thinking about and working on for a long time. And I think the short answer is there are definite, there are definite differences in terms of how the vasculature interprets different types of hemodynamic forces. And we did bulk RNA sequencing on the endothelial cells from the LPA model as well, and there is just not nearly as many kind of transcriptional changes that we see compared to controls as when we look at the shunt endothelial cell. So clearly but there are some. So it seems like pressure, the addition of pressure, whether it's kind of in combination or in isolation, is definitely engaging like a wider variety of biologic responses at the endothelial level. We're working with a group down at UCSD actually, who are developing a bioreactor that can actually really precisely separate the different types of mechanical forces, so we can study that better. Right now, actually, it's very hard to have a rig that can separate out pressure and shear. They usually just push flow through at a higher pressure to generate higher shear. So it gets very muddied with the in vitro work that's currently published and widely available. So we're trying to look into that and understand that more.
Thank you.
Oh,
Jason,
no, I got wrong.
This is Ron. Along the same lines, experimentally, I'm just wondering whether you see different reactions using flows through the regular heartbeat, which is phasic, versus a ECMO type of more continuous flow design.
Yeah, that is a fascinating question, and again, something we are wanting to study and will be able to, right? So we can also change, with these sort of newer devices that we're developing, pulsatile versus kind of continuous flow patterns to look at that. And then, I think where that's also gets very interesting as well is in the Glenn circulation and the Fontan circulation, obviously making the transition from a pulsatile flow, to kind of a much closer to sort of static and continuous flow, and what effect, that change has in conjunction with the changes in pressure and shear, is very interesting. And so that's something we're trying to develop as well going forward.
Thank you, Jason. Are there any other questions, comments? All right. Thanks so much for joining us. We'll see you all next. Oh,
yeah, I just wonder one way.
Go ahead, Paul.
Excuse me. Wonder if one way of a model that might separate out pressure and shear is to take a branch pulmonary artery and put a marked band on that branch and compare. So there would be little flow going through that one. You would see the pressure, but it would not see the flow. And compare that, the vessels, the endothelium in that branch versus the other branches.
Yeah,
a fairly distal band. So
Yeah, it's very interesting. I think that we're um, kind of developing some new models, and I think that definitely, developing that to be kind of like a reproducible in terms of like the pressure and flow stimulus that we're delivering would definitely take some model development, but I think would be very it would be very interesting as well to try to again to try to get at some of these very long-standing questions.
Yeah, I wonder if that would be similar to the kind of physiology we see with development of pulmonary arterial disease with acquired pulmonary vein stenosis, like what we see in the BPD population, and they have the redistribution of blood flow and develop arterial vascular disease in association with that. It'd be interesting. All right. Well, thanks, everyone. We'll see you in a couple weeks.